Hello and welcome to this video on the possibility and implications of trihelical DNA. As you may know, all known DNA is made up of a double helix with two matching and complementary strands of nucleotides. In some pop culture, new species are said to have DNA of unusual size, shape, and composition. An example of this is both the next logical step in complexity of structure, but also a perfect example of the problem, that is, trihelical DNA. As briefly touched on, all known DNA that has been a product of evolution and subsequent natural selection pressures is made up of a double helix, bar those that are an anomalous mutation, pathogenic, or normally an ad hoc solution applied temporarily. This is made up of a series of 4 possible nucleotides or building blocks that always occur in matched pairs. These are either a combination of guanine and cytosine or thymine and adenine. These occur in a sequence that corresponds to a gene. A gene is a blueprint for more complex products. The genes are used as a basis of protein expression, cellular regulation, and more. This occurs by opening the double helix and treating the 2 opposing nucleotides like a binary sequence. This then leads to a code for various products or events. This only occurs when read as a single sequence from one side of the double helix. When in a double helix, this form is unable to be used in any way. The double helix, when closed, is unable to be read. What then is the point of the double helix, you may ask? The answer is surprisingly simple: efficiency. A double helix is able to be stored in less space than a massive spool of single nucleotide chains. Think of what happens to your headphones, but on a far larger scale. On the other hand, a double helix is also more stable, consumes less space, and is harder to interfere with. This can be compared with a more complex structure like a trihelix. This triple-stranded DNA is much like the double helix in form and, theoretically, in function. It involves matching one nucleotide with its pair, in this case 2 matches. The method of creating could and should be similar to that of a double helix. The problems are physical and mechanical for the most part. Can it be opened and read with similar machinery to that used in a double helix? Would replication be easier? What of other advantages or disadvantages, such as being able to hold more data? To function, this trihelix would need to separate out the 3 strands. Compared to the normal 2 strands, this is far more complex. The complexes needed are not readily available to open up the helix to begin with, and adding in extra strands that need to be separated separately to the normal 2 methods is even more complex again. These strands then need to have both complementary components and a way to make them match in the context of competing with the other 2 strands. The bases of cytosine and thymine are pyrimidines, and adenine and guanine are purines. This has so far had the advantage that little effort is required to pull the 2 halves apart for replication, and this allows for the DNA to be copied or transcribed as needed. If you were to have another strand that matched these, then you would have more difficulty in separating them if they did not become bound up together. When DNA coils up to form a double helix, it is a very tight coil, and it requires little energy to stay in this shape. Another configuration would likely require more energy, and therefore is nowhere near as desirable. It would impart no selection advantage whatsoever. then you need to think about the cellular machinery required to open up and replicate or transcribe the double helix. It's already a very complex process, requiring many moving parts that change out as required. Then it needs to be able to match, fix, and or read the DNA along the way. Despite all of this, and the fact that it could be done, you would gain nothing from this third helix if there were only 4 nucleotides, as you are only adding a copy of the existing other complementary strand. This creates a significant amount of redundancy for something that is otherwise unnecessary. Even then, for this to happen, fundamental changes must occur in the nucleotides and helix structure. This is called a Hoogsteen hydrogen bond. It involves the thymine creating a hydrogen bond with the adenosine of the original double-stranded DNA. This creates a thymine adenosine and then an unusual bond to another thymine. Under other conditions, and in particular acidic conditions, cytosine can become protonated. This gives it a slightly positive charge. This then allows it to create a cytosine guanine pair with another cytosine with a special charge attached to that again. Of these 2 possible formulations, the thymine adenosine thymine is the most stable, whilst the cytosine guanine cytosine positive charge is also nominally stable by comparison to a straightforward thymine adenosine guanine and cytosine guanine guanine type triple bond. This bizarre triple helix structure was shown to exist a while ago and was found to inhibit the expression of a number of genes in certain systems. This is not to say that a triple helix does not serve certain or other purposes. In the current medical environment, it has potential as a therapeutic agent. The ability to target specific genes to modulate their structure and or function has far-reaching implications, particularly in the current biotechnology environment, where genomic medicine is becoming more and more possible. Take the example of sickle cell anemia, which is generally an inherited condition. It involves a specific mutation in a certain gene. In theory, you could have a third helix bind to the very specific genes by having it follow the same molecular matching pattern, and then interfere with the expression in such a way that you would then be able to resolve the sickle cell anemia. This would not be an unusual thing in nature. The triple helix does occur in limited circumstances, but this is nearly always restricted to plants expressing certain genes in certain ways at particular times. There is a strong possibility that a triple helix is common due to biological function, such as the DNA strand replication process or pathologies. There may even be reason for it to develop independently under different selective pressures, but it is not common in normal circumstances because it has no demonstrable advantages for a living organism that we know of. Added to this is the added cost of replicating a third strand of DNA that is otherwise redundant. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.